Hello everyone. The topic of my project was assessment of changes in the breathing rhythms after application of deep brain stimulation on the subthalamic nucleus of the right brain. The disease that I was interested in for my project was Parkinson's disease, which is basically a neurodegenerative disorder that is caused by the progressive loss of dopamine in human brain, and that basically leads to the tremor, rigidity, and loss of coordination in human body. And there is a well-established treatment that is being used for treating the motor symptoms of PD patients today, and that is deep brain stimulation. And especially the region that is targeted in human brain is subthalamic nucleus, or STN, which is a small region in basal ganglia that is responsible for regulating the human body's movements. Although DBS is very successful in treating these motor symptoms, however, it is causing another non-motor symptom, and that is breathlessness, and we don't know why is that being caused after DBS treatment. So in this project, I was basically interested in applying the DBS on the healthy rat's brain, especially targeting the subthalamic nucleus of the brain. And for that, I did 20 trials. I dedicated each 10 trials to each frequency, that was 10 hertz and 130 hertz, and these basically frequencies lie in the spectrum of lower and higher frequency range that are being used for DBS stimulation in Parkinson's disease. And I used two intensities that were 150 microampere and for 50 microampere for each frequency. And because the stimulation time was almost for 40 seconds, so I was interested in the 40 seconds right before the stimulation occurred, and then the 40 seconds of stimulation, and then the 40 seconds right after the stimulation stopped. After taking this 120 seconds period, I detected the peaks in time domain. I calculated the breathing rates from the peaks, and then I displayed all the 20 trials on one graph to see what is basically happening. So interestingly, if you can see in this graph, you may see that except for the red peaks, which basically refer to the highest intensity that was for 50 microampere and highest intensity that was 130 hertz, the rest of the trials and the rest of the intensities and for 10 hertz and 150 microampere intensity for the 130 hertz, we do not see any significant change in breathing rate, especially during stimulation. However, if you look at the red peaks, that basically shows that the highest intensity and frequency may cause change in breathing, especially during stimulation, and this is basically two times the rest of the breathing rates for all the rest of the trials. For next study, I would like to extend this project, especially by targeting the real PD rat model, so that we can have better assessment of what's basically happening after the DBS stimulation. Thank you.